Hi, welcome everyone. Thank you so much for joining us for this episode of Smithsonian Science How. I'm Maggie Benson. We're really lucky we were joined today by invertebrate zoologist Dr. Ellen Strong. Ellen, thank you so much for joining us.
Thanks, Maggie. It's great to be here.
So Ellen, you're an invertebrate zoologist here at the Smithsonian's National Museum of Natural History, and you've brought an awesome collection of things to talk about today. Can you tell us a little bit about your job here and the collections you use?
Yeah, so my official title here is research zoologist, and I'm a specialist who studies snails. And I also, one of the jobs that I have here also is taking care of our nation's snail collection. And with 15 million specimens, that's a pretty big job.
15 million, that's huge.
It's big.
So what made you become a scientist that studies snails?
So when I was a kid, I had no idea that studying and taking care of snails was a job description. I had no idea that this would be my career. I actually wanted to be an archaeologist, and I would take my dolls and bury them in the mud and carefully excavate them with my dad's tools, which he was not very happy about. But all that training went to waste because when I went to college, I wrote my undergraduate thesis on snails, and I was hooked from then on.
Very cool. So I see a lot of snails here out on the table. Can you tell us exactly what a snail is? There's a lot of variation here.
Yeah, so the technical term for the class that includes snails is called gastropoda. And this means in Latin, stomach foot. And so this refers to some of the main features of a snail. So you can see there on your screen the large foot that snails use to crawl around on. You can see that they have some tentacles with eyes attached to the foot. They also have a spirally coiled shell that contains all of the internal organs, including the stomach, and a trap door that closes behind them when they retract into their shell for protection.
I see some of these don't have a shell. Are they still snails?
You're absolutely right. Not all snails have shells, so that was sort of a trick question. Some snails have lost their shells, and this includes slugs that we see in terrestrial habitats, and it includes brightly colored nudibranchs that we see in the sea.
Now, what about how they eat? Do they have a similar way in which they do that?
Yeah, a lot of snails have a structure that we call a radula, and this is a flexible ribbon of teeth. And you can see this here on your screens of a snail extruding its radula through the mouth. And this flexible ribbon of teeth is used to graze algae and other substances that snails feed on.
Very cool. You don't usually think of a snail having teeth.
No, you sure don't. But there are some close-up pictures taken with a scanning electron microscope of what these teeth look like in close-up.
Very cool. So how many species of snails are there? With 15 million collection objects, there must be quite a few.
Yeah, there are a lot of snails. Most of the diversity of snails are marine. So there are about 36,000 species of marine snails that have been described. But snails, as you probably know, have invaded a lot of different habitats. So they've invaded freshwater and terrestrial habitats on all continents except Antarctica. And there are about 25,000 species of terrestrial snails and about 4,000 species of freshwater snails that have been described.
So terrestrial meaning living on land.
That's right.
So these shells over here look really familiar to me. They look like something found in the ocean.
Yeah, you're right. These are some beautiful examples of marine snails, what people would commonly recognize as seashells. And you can see that they are brightly colored in a lot of different shapes and sizes.
Yeah, that variation is really striking. I mean, here there's no spiral on the shell, and some of them are even completely different shapes.
Yeah, so some snails have taken this basic idea of a spirally coiled shell and modified it in ways that you can't even recognize it's a snail anymore. So in fact, this is an example of a snail. It's a group of snails that live together in a colony, and each one of those little tubes hosts a tiny snail.
Wow, that's incredible. It looks like a piece of coral.
It does.
So what about size of snails? A lot of the specimens here are really big, but is this the norm?
This is not the norm. I like to show seashells because they're something that people recognize, and they're beautiful, of course, but they are not really representative of the diversity of snails. So snails can range in size from a sand grain all the way up to more than 2 feet in length. So, and in fact, the large snails are in the minority. Only 10% of the diversity of snails are what we would call macromollusks, whereas 90% of snails are what we would call micromollusks and are only 4 centimeters in size or less. So 4 centimeters is only about 1.5 inches. So the vast majority of snails are, in fact, pretty small, and they are harder to find and harder to study.
So what about the snails that you study? What group do they fall in?
So they are not marine snails, but they fall into this group that we call microsnails. And so I actually study freshwater snails. The snails that I study are in this group of microsnails that are in about the inch and a half size range. And as you can see on your screen, they are not as beautiful, not as charismatic as their marine relatives. Not as highly variable. They usually come in shades of brown or green or yellow. And so they are not quite as charismatic as their marine relatives, but some of the features of the animals are really quite beautiful when you look at them close up.
So because they're part of this micromollusk group, this group that's very large and, as you said, a little bit more understudied, do we have a lot to learn about the freshwater snails?
Yes, there's a lot to learn. As I mentioned, there's about 4,000 species that have been described, but this might just be the tip of the iceberg. There are new species that are being discovered every day, and we estimate that 4,000 species might only be half of the number of species that there are left to document and describe. So we also don't really understand very well their roles in ecosystems, their ecology, their physiology, their anatomy. There's lots we don't know. And in fact, snails are really important in the ecosystems where they occur, and they are understudied relative to their ecological importance.
Interesting. So it sounds like there's a lot of work to be done.
There is.
That point about their roles in the systems that they belong actually is leading me to my next question, which is, what are their roles in these freshwater systems? But before you tell us, let's actually give our viewers a chance to weigh in. What do you think?
Sounds great.
Viewers, here's an opportunity to participate in a live poll with us. You can respond using the window that appears to the right of your video screen. If you don't see it, try minimizing your window to see the poll. Tell us, snails are important in ecosystems for making energy from sunlight, they're not important, preying on other animals, or being eaten by other animals. Take a moment to think about it and put your answer in the window that appears to the right of your video. So we're looking at all of your responses coming in. Ellen, we can see together that 81% of our viewers, the vast majority, think that their role is being eaten by other animals. What do you say?
I think that we have some very knowledgeable viewers here today. I'm really happy to see that nobody chose they are not important, because we know that they are. So I'm very happy to see that nobody picked that answer. That was sort of a trick question. But basically, all the other answers were right. They are very important in their ecosystems for food. They transform energy from the sun, and they actually do also prey on other animals. But not many people pick that answer, and in fact, it's somewhat rare. So I would say that answer is proportional to the occurrence in nature.
Now, if they are an important food source, what are their populations like in these freshwater systems? Are there a lot of them?
Yeah, so snails can occur in really high abundances in the communities where they occur. So as you can see in this picture, all those little dots there on the rocks are freshwater snails. And in fact, a good indication that an ecosystem is healthy is that it supports a healthy population of freshwater snails. And so they're playing, they can reach actually incredible densities in freshwater ecosystems. They can reach more than 1,500 individuals per a square meter.
Wow.
Yeah, that's a lot of snails.
That is a lot of snails.
And they are, They represent a very important component of the biomass. So they can represent up to 90% of the benthic invertebrate biomass in freshwater ecosystems.
That's incredible. So what animals are feeding on them if they are present in such huge numbers and represent that huge niche in that system?
Yeah, so most of our reviewers responded that snails are functioning in their ecosystems as food, and that's absolutely right. And actually one of the other answers was that snails are transforming energy from the sun. So we can see here in this simple food chain that snails are very important players at the bottom of the food chain. So they are consuming algae, and with a lot of snails, you eat a lot of algae. And so algae takes energy from the sun through photosynthesis, and snails then convert that energy into something that consumers higher up in the food chain can then make use of. And we see that in this food chain, we have fish and leading all the way up to humans. And so, in fact, humans are directly linked to snails through the food chain.
So what about water quality? Are they having an impact on water quality if they're in those systems in such abundance?
Yes, so one of the features that I did not mention that many snails have is a gill. And they are using the gill to extract oxygen that they use to survive from the water. And this filtering action actually has a beneficial impact on those ecosystems. They are filtering out harmful particles like sediments, bacteria, parasites even, and harmful algae, and keeping those things in low concentrations in the freshwater ecosystems where they occur.
So freshwater snails are keeping our water clean, and they're even feeding us through the food chain.
Yeah.
So they're really important for human well-being.
Yeah, they have a major impact on keeping our freshwaters healthy.
Does that go hand in hand with also experiencing some of the negative impacts that humans have on our freshwater sources?
Yeah, so you can think of snails as being surrounded in this medium in which they occur. And so they are in constant contact with water, and anything that humans do that has an impact on those freshwaters will have an impact on the health of those snails. So they are affected by the fact that we use freshwaters for agriculture. They are affected by the modifications that we make to natural rivers by damming. They're affected by pollution, habitat loss, mining. These are all things that are degrading our freshwater habitats and have a direct impact on the health of freshwater snail populations.
You mentioned that the snails are surrounded by freshwater all the time, and they kind of occur in these small areas where there is freshwater. Does that impact their vulnerability in instances where we may have a negative impact on the water source?
Oh yes, absolutely. So you can think of, so freshwater snails are reliant on being in water for their survival, and they can't just pick up and move when things get bad. And so they have to adapt to the changing habitats where they occur, or they're driven to extinction.
Does that make freshwater species of snails more susceptible to these changes compared to their marine relatives?
Yes, absolutely. So you can think of the way that freshwater habitats are distributed compared to marine habitats. So the marine oceans are more or less continuous habitats, but freshwaters are not continuous across the landscape, and so freshwater populations are not continuous across the landscape. And in places where we have had an impact on those freshwater populations, those populations have declined in number and in size, making them less resilient to human impacts. And we see that in the numbers. The numbers speak for themselves. So 177 species of freshwater snails have gone extinct, whereas only 6 species of marine snails have been documented as being extinct.
Wow, that's incredible. So we really need to be paying attention to our freshwater sources and the snails that live in them.
We do.
We have a ton of questions coming in, so we're going to learn more about your research in a moment, but let's get to some of those questions.
Sounds great.
All right, this question comes from MacGyver. MacGyver asks, why do you study snails?
I study snails. Yeah, MacGyver, that's a great question. I study snails because there's so much to learn. Snails are an incredibly diverse group, and as I said, they are understudied relative to their diversity. So almost everywhere you look, there is something new to learn. Every day I come to the office, I find out something new about snails. And so it's sort of like Christmas every day.
Ellen, let's learn a little bit about your research. You started telling us a little bit about the negative impacts that humans are having on freshwaters, which is in turn impacting snails. Can you tell us about how the snails you study have been impacted by humans?
Yes, so one of the groups of freshwater snails that I work on are called pleuriserids, and we actually have some examples of pleuriserids here on the table with us today. Pleuriserids are the second most diverse family of freshwater snails in North America. There are about 160 species of them. But they have been heavily impacted by human activities. And so all the threats that we talked about, damming, pollution, habitat fragmentation, and here you see a nice picture of a large hydroelectric dam. These have had a major impact on our freshwaters and on the diversity of pleuriserids. So you take a free-flowing river like this and you put a hydroelectric dam on it, and this has been the source of major extinctions in the United States. So in the Coosa River in Alabama, this is a river that's about 280 miles long, and a series of dams that was constructed on this river basically took a nice free-flowing river in its natural state and transformed it into a series of lakes.
What did that lead to?
That led to the biggest extinction of freshwater organisms in the United States history.
Wow.
And this also led to the extinction of some pleuriserids. So
is that what we're looking at here, some of the extinct species?
That's right. These are some of the extinct species of pleuriserids. So already 33 species and one whole genus of pleuriseriid has gone extinct caused by these impacts.
Wow, that's really jarring considering how important freshwater resources are for every plant and animal.
Yes.
And here are more of the snails that are at risk?
Yes, so even the snails that have survived these extinctions are still at risk and heavily imperiled. So out of those 160 species, 80% are experiencing some level of risk. So this ranges everywhere from slight impacts all the way to being on the verge of extinction. So around 130 species of pleuriserids are experiencing some level of risk.
So what does studying these snails actually contribute to the overall understanding of them? But actually, before you tell me, let's again ask our viewers for their opinion.
Okay.
Viewers, here's another opportunity to participate in a live poll. Tell us, what does studying snails help us understand? Individual species, snail biodiversity, ecosystems, or food webs? We're again watching your results come in. Ellen, we can see that people have responded, given responses for all answers, but mostly snail biodiversity. What do you think?
Yeah, great. There was no trick question here this time, so these are all right answers. As I mentioned earlier, there's still so much to learn about freshwater snails that there are things to learn in almost every area. So learning how individual species interact in their environment, how they contribute to the food webs where they occur, their ecology, their physiology, these are all things that we need to learn about. But the most popular answer, which makes me very happy, is understanding the biodiversity. And that's my job, basically. My job is to document and describe the species of freshwater and to understand their biogeographic distribution. So
that's really foundational for understanding everything about these animals and even how to protect and conserve them.
Yes, absolutely. So we can't conserve and we can't protect what we don't know. So my job is to make sure that we document and describe every species and that each one has a name. And this is the basis for communication. Species are really the currency of biology and conservation biology. And that's where I make my impact.
So how are you documenting this diversity? Are you using a selection of the museum's 15 million collection objects? Are you going out into the field? Do you do both?
I really do both. Museum collections are an integral part of what I do, and the reason they're so important is because, and as you can see here, these are just some of those objects that I help to take care of every day. These museum collections help to document the distribution of species, both past and present, and they are repositories for undiscovered biodiversity. But getting out in the field is also really important so that I can see how species vary across the landscape, where they're distributed, what their ecological preferences are. These all help to inform our interpretation of how many species there are.
Do you need special gear when you go out into the field?
I don't really need too much special gear for the species that I study. They're mostly in very accessible habitats. Sometimes, as you see here, I'm working in places where it's an active mining area, which is why I had that helmet. But mostly, I just need a GPS unit and or a couple of helpful colleagues.
Those are the tools that I think I would like to use collecting them.
You just need a couple of friends with you in the field.
So when you're out in the field and you find a species, do you sometimes just know that it is brand new and nobody's ever described it before?
Every once in a while, you will have that amazing aha moment in the field where you have found something that is so unusual, is so far away from everything else where you would expect it to be, that it has to be new. But for the most part, most of our discoveries come in the lab. And really the reason is because shells, which is what you have access to when you're in the field, are not really a very good indicator of species diversity. We have a couple of specimens here to show you that really shells can be quite variable within species. And if this is all that you have access to in the field, this is not going to be a reliable indicator of what you have as new.
So what tool do you use to determine if it is a new species or not?
So we preserve those specimens in the field, usually in alcohol, or we freeze them, and we bring them back to the lab. We take a small piece of their tissue and we sequence it, and we get their DNA sequences, and we compare many DNA sequences from many individuals for those species that we're studying.
Which we're seeing here. So
That's
has
right.
this DNA analysis helped you discover new species?
Absolutely, but not just new species, whole new families.
Wow.
Yeah, so some of the species that I work on occur in the Pacific Northwest. These are species that used to be called pleuriserids. This is in the genus Juga. And we were able to compare DNA sequences of Juga from Washington, Oregon, and California, and you can see some of the diversity here, to other species of pleuriserids, and we found that they weren't close relatives at all, that in fact Juga is most closely related to species in Asia. And we united them in their own family, the Semisulcospiridae.
That's quite a mouthful. I'm not going to repeat that. I think I'm going to stick with Juga.
I'll stick. Me too.
Were you able to describe new species within this brand new family of Juga?
Yes, we found several new species. One of the species that we found are those specimens that you see there in those vials. Those are individual sequence specimens of a new species of Juga that we found in eastern Central Oregon. And this is one of those species that, unlike most species of Juga that occurs in rivers and springs, rather rivers and lakes and creeks, this is a species that occurs there at that orange dot in these isolated springs in meadows like this. And there's my colleague helping me to collect some of those specimens. And we brought them back and we sequenced them and we found that they are a new species.
Do we know what the name is yet?
So actually these results are so new that we haven't had time to give it a name yet. We're still working on that.
So given its isolated location and not finding them anywhere else, and all of the threats that you have been talking about, is this a little bit of a race to name this given the threats that face freshwater snails?
Yes, absolutely. So this is a species, as you said, is highly isolated, and that makes it especially vulnerable to human-mediated threats. A single catastrophic event could wipe it out even before it has a name. So we really are in a race to catalog and describe these species, let alone understand how they function in their environment.
So it's not all doom and gloom, though. There are some restoration and recovery efforts underway that your research has helped inform. Can you tell us about those?
Yeah, so there are a lot of efforts underway to help improve the quality of our freshwater ecosystems. So one of those efforts includes dam removal. We've talked a little bit about the impacts that dams have on freshwater ecosystems in fragmenting natural populations. You can see here that this has a beneficial impact on restoring rivers to their natural state. And these are some of my colleagues at the Alabama Aquatic Biodiversity Center who propagate freshwater snails and return them to these communities where they used to occur and where freshwater health has been restored.
Ellen, this is another great example of how your work is really foundational to the future of freshwater snail species and the restoration and recovery efforts. Thank you so much for helping us learn more about your freshwater snails and your research here at the Smithsonian. We have so many questions and we're a little over time, so we're going to get to as many as we can right now.
Sounds great.
All right, this question comes from, I just lost it. There are so many coming in. Addie had asked, let's see. Or Addie, send your question in again so I can see it. But this one is from Emma and Sophia. They want to ask, are snails threatened?
Yes, snails are highly threatened. So freshwater snails are among the most highly imperiled animals on the planet and have experienced the highest numbers of extinctions. So freshwater snails are very important, but they are heavily impacted by degradation of their habitats and human impacts and have experienced large numbers of extinctions.
So Bella and Sienna ask, what would happen if all snail species went extinct?
Oh dear, I don't even want to think about that. That's a very depressing thought.
No, our poor freshwater.
Our poor freshwater, yeah. As we've talked about, their role as ecosystem engineers means that they play vital roles in maintaining our freshwaters and at the base of the food chain. So you can imagine if there were no more freshwater snails, some of our food chains would collapse, and these are food chains that we rely on to provide us food.
This one's from Jordan and Mariella. I saw snails in my backyard after rain. Why?
Well, that's also a great question. A lot of snails are, even if they don't live in water, are reliant on water for their livelihood. So some snails, some terrestrial snails, when conditions are dry, they will retract into their shells, they'll bury in the ground in order to preserve moisture. And then when it rains, they can come out and crawl around, feed, reproduce, do all the things that snails do.
Ellen, thank you so much for answering all of our questions. Viewers, thank you so much for all of your awesome questions. I'm really sorry that we didn't have time to get to all of them. We're really over time. Ellen, can you tell our viewers where they can learn more about your work? I think they're all really interested.
Sure. Viewers can go to Conservation International or the Freshwater Mollusk Conservation Society to learn more about freshwater mollusks and conserving our freshwater resources.
And we have resources on the Curious website, curious.si.edu, about Ellen's work and freshwater snails. So Ellen, thank you so much for being here. It was a really great show. And viewers, thank you so much for tuning in to this broadcast of Smithsonian Science House.